Okay, welcome everyone. Good evening. Thank you for joining us for today's Café with Jagesh Shah and Dr. Melanie Ivarsson. Before we begin, I want to take a moment to thank the Café Library Foundation, whose mission is to raise private funds for the library. Their support enables us to bring programs like tonight's event to you. The foundation plays a unique role with the Science Café series and also produces the content for these programs. Jagesh Shah will be your host for this evening. Jagesh is a distinguished scientist with a background in biology, computation, and engineering, who is also a Lexington resident, former Carey Library Foundation board member, and the creator of the Science Café series. Jagesh, welcome. And now I'm going to hand the evening off to you to host and introduce our special guest for the evening.
Oh, thank you, Helen, for that introduction. So welcome everyone to the Carey Science Cafe. I'm Jagesh Shah. I'm, as Helen said, local Lexingtonian and your host for this evening. For those of you new to the Science Cafe, we bring in, scientists and biotechnologists from the local Lexington environment, which, not difficult to find, but given the environment, but really an idea, our goal is to really try to figure out what they're doing, how they got to where they are, and any advice that they can provide for us about their role in bringing science to the masses. Today we're very lucky. We're continuing our COVID conversation discussion. So last April, we spoke with Mike Keneally, the Mass Secretary of Housing and Economic Development, who was also the co-chair of the Reopening Advisory Board, and he provided a great environment to discuss kind of how state government was ensuring that we weather this pandemic. And today we're really taking a deep dive into the medicines that are tackling the pandemic itself. We're honored to have Dr. Melanie Ivarsson as our guest. Melanie is a Lexingtonian, distinguished scientist, but most relevant for this evening, she is the Chief Development Officer at Moderna Therapeutics, a biotechnology company based in Cambridge. Many of you will have heard that Moderna is developed one of the leading vaccines against SARS-CoV-2, and they've been a real beacon of innovation in Massachusetts. I would be remiss if I did not also mention that Melanie has recently been recognized with Order of the British Empire. And that is a distinguished honor that I think really underlies a great deal of her role in really staving off, the impact of this viral pandemic. So thank you, Melanie, for joining us tonight.
Thank you so much for inviting me. That was an amazing introduction. Thank you.
Oh, well, I'm sure that there are many more to come, and I'm glad I can be of service. One of the things that we generally do in the Science Cafe is we'd like to start at the beginning, kind of, your origins, as it were. Where did you grow up, and how did that early interest in science develop?
Absolutely. Yeah. So I grew up in the UK, and I was very fortunate. Neither of my parents actually went to college. They were sort of post-World War II sort of impacted teenagers at the time. And neither of them had that opportunity. And for me, they thought education was unbelievably important. And so they sent me, and we have quite a lot of segregated education in the UK, and they sent me to an all-girls school in Hampton in Middlesex. It's one of the oldest girls' schools in the country. It was founded in 1711. And it had some fairly archaic principles. The headmistress had to be female, over 35, Church of England, all these kinds of things that they don't do anymore, but when I was there. And one of the things that was the best thing for me was we had a headmistress who had a PhD in chemistry, and she came into the school and basically revolutionized the school and built this enormous science block. So you had music and arts and languages and humanities and science all completely level within the school. Wow. So there was as much interest in each of these as each other. So there was no such thing as girls' subjects or boys' subjects. And this amazing new wing of the building, to me, just sort of piqued my curiosity. And so I ended up loving my science classes all the experiments. It was so much fun. I had so much fun with my friends in those classes, and ended up doing sort of science A levels, which is what we do in the UK, in maths, biology, and chemistry. And that really sort of sent me off into the pathway of science. And so when I was thinking about what to do at university, I had a real interest in sort of the human body, and ended up doing a degree in physiology, which was a sort of a modular degree. So you would do a bit of pharmacology, a bit of psychology. You could choose, much like modern degrees are now. Modern makes me sound so old. But the shock to me was at 18 years old was the first time I was exposed to the fact that STEM was apparently boys' subjects, and the girls were all down the road doing sort of science, doing languages and humanities. And at that time, the science subjects were still very sort of male dominated. And it was really interesting, because I think I was very fortunate in the fact that I had not had any of those influences all the way up to 18. Did my degree in Bristol. And, I think to sort of follow the story through, really loved what I was doing, loved the practical application of science, really enjoyed neuroscience. And one day I was doing an experiment in the lab, and my undergraduate sort of project supervisor walked in and said, "Have you ever considered a PhD?" And I was kind of like, who are you asking? And he was like, "You." And I went, "Really? You think I'm good enough to do a PhD?" Because I thought you had to be kind of like some amazing kind of academic. And he was like, "Yeah, you can, like, let's get a grant for you. And I think that started the journey for me of the importance of mentors in your life, people who maybe believe in you and see things in you that you don't see yourself. And stayed at Bristol to do my PhD, which was in neurophysiology. Which I loved, really enjoyed that. I was very fortunate to meet my husband while I was doing my PhD. An example of male-female ratios, we met at a cerebellar conference. There were 124 men and 4 women at that conference. So, he's just very lucky that I picked him out of all these opportunities. All joking. And then sort of moved to New York to do a postdoc after I'd finished my PhD. And then I went to Lund in Sweden to do another postdoc. And it was around about that time when I thought to myself, I think I was at 4 o'clock in the morning, I was running an experiment, and I went, I have run out of interesting questions to ask. And I thought, oh dear, I'm 26. I've got no more interesting questions to ask about the cerebellum. I need to leave this to somebody else, and I need to go and find something else to do with my life. And so we moved back to the UK, and that's when I moved into industry. The application of scientific principles in clinical research just really drew me, and that started my 20 odd year.
Wow.
No, I was 20 odd year sort of time in industry, in clinical development.
Wow. So that transition is, certainly, I think for myself as well, that doing it right out of a postdoc is, in academia, to kind of lift up the rug a little bit for people, wasn't always considered a good transition. Right. And, but I think, what was your experience in making that transition into industry?
Oh, absolutely the same. I remember feeling apologetic and knowing that I'd have to go and tell the academics in my life that I was selling my soul to the devil, which is how they saw industry. And, think how far we've come. This is the late 90s. My first job in industry was in 99. Think how far we've come now, where that industry academic partnership, which so exemplified actually by the COVID-19 vaccine development, right, is where great things happen. It's not about it, academia has the upper hand. We need each other. That partnership and collaboration is so crucial, and that's where amazing things happen. but you're right, 20 odd years ago, it was really frowned upon because that was where people went to make money, and they had a completely different set of values and they made decisions completely differently. And of course, it wasn't like that when I got there at all. It turned out that people do amazing science within biotechs and pharma companies. And there is some really amazing sort of basic research done in those environments. It's not just all about looking for the next drug candidate. There is a huge amount of amazing research done everywhere. And those partnerships are what's really sort of been an inflection point, I think, for the industry.
Yeah, and just to kind of reiterate, what I have seen is really, particularly now when there's a remarkable amount of opportunity for investment, academics are taking their early science and driving, the vacancy rate for available space in a place like Cambridge is essentially zero, right, at this point. And so we have a lot of science transitioning. But presumably you got a chance to try a lot of different kinds of science out, and over the course of your career, you're making more than cerebellar drugs. But
Yes.
did you start out in neuroscience as a kind of the thin edge of the wedge, and then transition into a variety of different areas? Because now you're working on clinical stuff, right? Like at least in part. So that's quite a move too.
Yeah, so my entry into the pharmaceutical industry was through the very early parts of clinical research. It was the translational medicine to early clinical group. And that's a really good place, I think, to enter the industry. If you've had an interest in research, you can go either way. And so I moved to Eli Lilly in the UK and was in their clinical pharmacology group. And in those days, when you were setting up a study, you did everything. You designed the study, you wrote the protocol, you set it all up, you did the contracting, you did the whole thing, you analyzed the data, you wrote the report, you did everything. And it was the most amazing foundation in sort of clinical research. And I just fell in love with it. I'm very proud of the fact that I've wanted to go to work every day my entire career. I just think clinical research is an amazing space to have a career. And there is so much you can do through, whether it's small molecule or large, vaccines, therapeutics. No 2 days are the same. And there is opportunity within, all of industry, but in clinical research, there was a real opportunity to work my way through the different phases of development. So most of my work at Lilly was in early phase research, clinical pharmacology. Then we moved to Pfizer. Both my husband and I moved to Pfizer, and I spent 5 years in the UK there, primarily again in early clinical development. And then we moved to Sweden for a couple of years, and I stayed with Pfizer and moved into later phase clinical research. And then when we moved back to the US with Pfizer, I did a strategy and operations type role, which really gave me insight into the finance and business planning and project prioritization. And then went to Shire here in Lexington. Best commute ever. And I really wanted to spend some time in rare disease. I thought that was a really interesting space to be. And that was, I absolutely love Shire. I thought it was an amazing company. And I know many colleagues in Lexington are there as it is now. I'm under Takeda, and joined in 2015. And, Shire had a really good culture, really strong respect for sort of functional expertise. And we did a lot of really amazing things during that time. And then we were acquired by Takeda, which is also another great company. And I was very happy to stay at Takeda. And then one day got a message from somebody at Moderna going, would you be interested in chatting to the CMO of Moderna? And, you'll have to sort of bear with me here. This was November 2019. And I went home to my husband and said, who are Moderna? Have you ever heard of them? He was like, oh yeah, they're that mRNA company in Tech Square. I used to be in the same building as them. And yeah, they've been, doing that for about 8 years. That would be interesting, go for a chat. And I remember spending what was supposed to be a 30-minute sort of chat with the chief medical officer turned into about 3 hours. as he just told me all these amazing things you could do with mRNA platform technology. And this was a tiny little company that had literally, we were talking hundreds of people in the company just 2 years ago. I mean, there's only about 2,500 of us now, even with, having produced 1 billion doses of COVID vaccine. It's remarkable, really. But they'd run a few clinical experiments with this new technology. Every single clinical study could be the end. But they were starting to think about their first large-scale phase 3 trial in about 18 months hence for CMV. Would I have any interest in going and helping them build out their clinical development organization and be their chief development officer? And I sort of should also point out that I'd gone off and done an executive MBA at Sloan at MIT. And so there was me thinking, well, I've got this toolkit, I'm ready for a new challenge, but why would I give up this great job at Takeda to this little biotech? Because you can get stuck in biotechs and, it could be years before we ever sort of went into the clinic or did anything. And I remember they said, well, there are 32 people in your group, and over a couple of years, you could grow it to 55, but that would be about it. And I was like, well, the size of the group doesn't matter, but that gives me a sense of what I'm building. They've never run a phase 3. Could be interesting. I'll do it for 2 years. I'll go for 2 years and see what it's like. So I turn up on the 3rd of February, which is like day 1. And they go, so have you heard of COVID-19? And I'm like, yeah, that's that virus in China. Like, so we're thinking of, making a vaccine, not sure if it'll be needed, but, we're thinking of, now you're not to have anything to do with it. We're just going to run it as a little side project. You worry about the rest of the business and we'll let you know. And then, of course, 2 weeks later, it's amazing now when you look back at the timescale and how little we knew. It was, we're going to do this. And this is going to be everything we do. And we've got to go and work out how to go and run our first ever large scale trial, how to manufacture this, and we had no idea if mRNA technology would even be the solution in a vaccine. And that was the start of a very amazing 2 years.
So we're going to get into this because I think the last 2 years, what little you might be able to remember of it, because it's gone undoubtedly incredibly fast for you. I think we should think about that. One thing I wanted to make sure that we think about is that, it sounded, a lot of the work that you did as a postdoc and as a PhD student would have been very basic research. Mm-hmm. And you decided that you wanted to go to clinical research. And so maybe you could just, for our audience, really kind of play out, what's the fundamental differences? One is, where do you work and what are the kinds of core, critical questions that come up in each stage?
Yeah, no, that's a great question. And I'm sure there are, knowing the Lexington community, there are many people who know a lot more about all of these areas than me. But I think, in academic research, I think we all know that you have chosen the question you need to ask, and that becomes everything you are doing to support your grant and your publications, and you build a whole lab around answering that question, what your hypothesis is. And what comes from that is an amazing amount of incredible basic research that informs us about biology and chemistry and what we are doing. 20 years ago, when I was moving into industry was more about the application of that research. There wasn't so much what we've seen now, particularly in the Cambridge ecosystem, is this incredible entrepreneurial spirit within the academic environment that's then taken that to form biotechs. But 20 years ago, you had to really go into industry to see that research get turned into the start of drug development and then go into clinical. And so the transition from that basic research to drug development and then for me into clinical was all about, taking a molecule that you've designed and done preclinical testing into the human, and checking, first of all, that it's safe and tolerable, and then working at how do you test if it works, right? So if you've got a diabetic drug, you're going to obviously be looking at glucose and HbA1c and all those other things. If you're developing a vaccine, then you're looking at, does it prevent infection and what is the reactogenicity? So for those who've never sort of gone into clinical, those experiments are incredibly carefully designed, just like they are in the bench. And the participants, the people who've chosen to sign up to this are the heroes. They are the heroes of the story. They are the people who've said, yes, I will let you test your vaccine or your drug on me. And so you very carefully have to think about the questions you want to ask and set that up. And I think one of the things everyone should also be aware of is this is one of the most highly regulated industries in the world, probably the most highly regulated. I mean, anybody who works in clinical research, good clinical practice is your book, and you do everything within that framework. And there are very significant consequences for everybody if you deviate from that. And participant safety and data integrity are just those fundamental tenets by which we live our lives. And you do everything to protect those, the people who volunteered or the patients and the data that you generate. And from that, you are then able to collect that data and submit it to regulators around the world, and they will either approve your vaccine or drug or not. And that whole process fascinated me. And I guess maybe I just like the organizational aspect, I don't know, but along the way, I really enjoyed the teamwork. So for me, it was interacting in those cross-functional teams, being very involved with the projects and seeing them through. And one day, that drug going into somebody. And, I think for me, I remember being at Lilly and working on a diabetes drug and getting so kind of immersed in the team at the time of doing the submission that I missed my son's first steps. My husband was with him. I was on a plane. And I carried that guilt. I know he's watching actually, so I hope he knows this story. Otherwise, sorry, Mum wasn't there. And I remember some years later, my husband sitting next to somebody on a plane, and they got into a conversation. What did you do? Blah blah. And he, this passenger started telling my husband about how he'd just gone onto this new diabetes drug and how he'd got his life back and he'd lost all this weight and he had all this energy and he could now play with his kids, and he was so grateful. And my husband went, my wife helped develop that drug. And it kind of, that's when you start to think, gosh, there's an impact to what we do that is just so incredible. It's really, until Moderna and the vaccine, it was very rare to ever meet anybody that had taken a drug that you'd worked on, because that's not generally conversations you have with people. But every time I did, and I heard those stories, you can't help but think what we do is just so incredibly impactful on people's lives. And so it's an amazing thing to be part of. I think it's a real privilege to have the jobs we have.
I couldn't agree more. And I think, even on now what we see as more of the basic research that actually takes place in industry, because it's evolved over time. It's still a remarkable driving force for some of those early scientists to know that they are making medicines that will go into people. And for them, it's a much longer path. They are just starting at the bench, and then they're going to watch this thing move. But it keeps them going. It gets them up at night, as you described, or gets them up in the morning, sometimes at night actually too. And they come in every day energized. So it's remarkable to hear that, kind of you've got that enthusiasm and that patients are driving you, and that's exactly what we would want. So of course, kind of the one of the most, I think, for biotech, one of the most interesting times has been this last 2 years, to try to understand as a pandemic comes in and all the constraints that puts on our ability to do work, that Moderna was able to even develop and then manufacture and then distribute, kind of a vaccine. So I think we want to just kind of start at the beginning. I think, a little bit about what mRNA technology might be. And then why don't we talk about like what it was like to make that and, kind of those first clinical trials.
Sure. So mRNA is, we call it sort of, the software of life. And basically the basic principle is that you are turning your body into its own antibody factory. So you introduce something into the body that it creates antibodies against, and so it develops its own immunity against the infection. So that's the basic principle. And the mRNA vaccine essentially mirrors the spike protein of the COVID, the virus. So we all know what a COVID-19 virus looks like now. In fact, I have one somewhere here. So basically, my husband made this for me on the 3D printer. So, you never know when you need to show and tell. And so it basically replicates the spike protein. So you're not injecting the virus into the body. Some of the other vaccines, sort of take inert virus and other things, but you replicate this. You then wrap it up in a lipid nanoparticle and you inject it as your vaccine. And what that then does in the body is the body detects the sort of copy of the spike protein and goes, oh, I need to develop antibodies against this, and then develops protection for you. So that's a very simple way of looking at the mRNA. What Moderna had before I had arrived, Moderna had spent quite a lot of time already looking at MERS and other sort of respiratory viruses, and had established relationships with the NIH and the US government to start thinking about pandemic preparedness. And so they, when COVID-19 became very apparent that this was going to be a pandemic, there was a very early conversation of, shall we do this? And the answer is, of course, because we think we actually have a solution and a way of doing this. And so they went into the lab and they got the sequence and they started to create the mRNA. And they'd already Moderna has quite advanced LNP technology. And so they were able to generate from the sequence to shipping for the first clinical trial in 42 days.
So maybe we can give people a sense of like how incredibly fast that is. I mean, for example, your diabetes drug, like from the, what would be the equivalent? Would it be the first time you manufactured at scale, or, what would?
Well, I mean, we weren't even manufacturing at scale at that point. We were doing small batches for those early clinical trials. We were doing small batches. So I think, to give people there are many, I know many in our community who do this day in, day out, and there's a range within every industry, but those early development, you're talking years. You're normally talking years, you're identifying a candidate, you're finessing your chemistry, and then you're doing tox studies, which we didn't have to do because we already had proven the platform approach. And so we were able to, and this is this is why mRNA technology is so well suited for pandemic response, is because you can identify your mRNA and create a small batch and actually have it ready for clinical testing in less than a month and a half, which is just phenomenal. Most of the other vaccines took months to get to the same stage. And that's really what when it came to starting the clinical trials, that put gave BioNTech, Pfizer and Moderna that advantage, which is why those two companies were able to get their phase 3 trials started the earliest. And so you asked about the clinical testing. We actually shipped it off to the NIH, and they ran the first human trial which started in March of 2020. By May, we had started the phase 2. So we actually put it, you inject day 1, you inject the second dose a month later, you look at the safety, and based on that simple safety decision, we started our phase 2. We then, in May, started our phase 2, looking at several different dose levels, and in July, started our phase 3 study in 30,000 people. So the vaccines normally take 10 years. And it's very interesting when I hear people say, well, it normally takes 10 years, so I don't trust it because they did it in 11 months. It takes the same, you do the same things in 10 years. We didn't do anything differently or anything less. We did more things in parallel. So what you would normally do is you would take a couple of years to develop your vaccine. You would run your phase 1. You would then look at all your data just for weeks, not quite sure necessarily. The decision's the same, but we all look at it for a very long time. You then run your phase. Have we lost Jagesh?
Sorry, I am coming back.
Okay, I'll just keep talking. So then you run
This
your
is the
phase
modern
2.
era. It's like, you know, all your
Contactable all the
computer,
time.
all your phones and everything are connected. I'm very sorry.
No worries. You'd run your phase 2, which would take months. And then you'd have to make this massive investment decision about building a factory, which would cost hundreds of millions of dollars. And that would be a pause for thought. And then you would start doing that in parallel with your phase 3. And that would take 10 years. I've just told you, we did the development in 42 days. We did the phase 1 in 6 weeks. We did the phase 2 in 6 weeks. And we started our phase 3 using the exact same data that we would have done if we'd done it in 10 years. And that's the difference. And the reason is the upfront investment. We chose to go and build a factory at risk with the support of Operation Warp Speed. And we invested upfront in setting up our phase 3 trial at risk. And, you know, to put it in context, a phase 3 trial of that magnitude would normally take 6 months to set up, and we did it in 14 weeks, start to finish.
So I think that, you know, all these phases, you know, kind of, I think, you know, for some of our listeners, probably, you know, kind of, you know, the language and like that, but really that science, and I really think that, you know, it's a simplified way of looking at it, but somebody sequenced a virus, got a sequence, and Moderna was able to type it essentially into something and have that mRNA come out the other end. And I think that it speaks volumes around the preparedness around the platform. The fact that I think somebody asked a question about an LNP, lipid nanoparticle. Maybe you can give a little bit of view on that, but the mRNA sits in there, and really it's whatever sequence you type in that you can get in there. So you have, you know, this remarkable modular system that you were able to exploit. And that's really where the speed comes from on the science, on the development side.
Yes.
Right.
And the fact that we've done it many times before. And so everything is set up to replicate as a platform. The ability to generate the mRNA, the ability to manufacture, everything is set up. So it's a bit like in sort of baking. If you go to a bakery and everything is set up, you weren't starting going, what piece of equipment do I need for this? We have everything already set up, and we just put in the code and then press the button. It's not quite as simple as that. It's not
quite that. Okay. I don't
want to give anyone the wrong idea, but what I want people to understand is we've been doing this for nearly a decade. We'd never run a phase 3 trial, but we've been developing mRNA vaccines for nearly a decade. So this wasn't something completely new. That this has been something that academia had been looking at for 20 years. And so doing it at this pace and scale speaks to the pandemic, it speaks to the culture of Moderna, it speaks to the fact that, you know, you've got a small group of people incredibly committed to this. But this was a tried and this was a technology that we knew a lot about.
Yeah, and I think that's really important to point out, that the science could move fast. But as you point out, even if the science moves fast, when you now have to engage in clinical testing at scale, that's not something that that's unique to this pandemic. And I know that you guys went through a lot of different partnerships around the country, around the world, to be able to make sure that this could happen.
Yeah, no, I mean, you're absolutely right, and thank you for pointing out about the phases. And I come from a world of complete jargon, so I apologize. Let me quickly go through those. So the phase 1 is very small testing, really small groups of individuals, maybe 10 or 20 people that you give the vaccine to, and then you're looking to see basically reactogenicity or adverse events. Do they get a sore arm? Do they get a fever? Do they get a headache? How are they feeling after that, versus people who are given an injection of placebo or just saline into their arm? Phase 2 is you scale up a little bit. You go into a couple of hundred people at a couple of different doses, and you're really looking to see what you can only commit to one dose in your phase 3 in a pandemic. You've got to get that dose selection right. And we chose to go with 100 micrograms because we believed that, which is the dose that people will have had if they've had their primary series with Moderna vaccine, because we knew that it would be, that would give good coverage, and we hoped it would give durable coverage for a long time for people. And that was behind that selection. And then the phase 3, the large study, was in our vaccine versus just salt water, saline. And that was in 30,000 participants. So imagine a company that had never run large-scale clinical work faced with setting up a 30,000 subject study faster than probably anyone had set up a vaccine study. And that is in part because we have an incredible team at Moderna, and it isn't, companies don't run clinical programs, people do. And people were literally ringing us up saying, let me help, let me come and be part of this. I will, when can I start? I will start next Monday. I will give up my job and I will just, come and be on this journey with you. And we had some amazingly talented people who just put their hand up and came and went on that journey with us. And I'm incredibly grateful to them for doing that. And we partnered, as you said, we were under Operation Warp Speed. And so there were a few major differences when you're doing clinical development in a pandemic. Firstly, absolutely everybody in the ecosystem, whether it's, everything has to get reviewed by an independent ethics committee before it can go into human testing. That's really important. Normally, they would want 2 weeks to review a document. They're saying, we'll clear our calendars and we'll do it in 1 day. And so that 2 weeks becomes 1 day. The FDA were amazing. So I'm pretty sure everybody knows who the FDA are, but as regulators, they were working with us talking about the endpoints and what data they would want to see, and being incredibly collaborative to make sure that we could get a head start on preparing the data packages that they would be expecting to see. And then every single clinical trial site that we went to and said, do you want to be taking part in a vaccine trial for COVID-19? And they were like, yeah, absolutely. And then we were really fortunate that the NIH formed the COVID Prevention Network, which was a network of academic centers with really top experts in sort of vaccine development and infectious disease. And in our trial, we had about 30 of those expert sites. They actually worked across all the sponsors. So they worked with AstraZeneca, Johnson & Johnson, Novavax, some of them work with Pfizer as well. And so this network of sites plus what I would call industrial vaccine research centers, we had about 70 of those, and they were dotted all around the country. Now, as I've said many times, it was the first time Moderna had done this. So we had no operations outside the US. So we could only really run our trial in the US because we didn't exist outside the country. Nobody outside the US had heard of Moderna until COVID-19. And so we ended up with 99 sites in the US, all amazing, who enrolled 30,000 volunteers into our vaccine trial in just over 12 weeks, which is just phenomenal.
Well, and again, this is I want to point out two pieces of that I think we just need to drill down on. One is this idea that people put aside a good deal of their other workload to make sure that not only did the data get the right analysis, right, that it also you got the right expectation. You were given the right expectation as to what to give them. So the FDA didn't suddenly, kind of say, oh, we're just going to let this stuff go. They made sure that it, as you said, all the same data was generated. It's just a question of ensuring that, this was, I can tell you from my own experience, COVID really did slow down the FDA for many other things. And appropriately so, right? Is that the prioritization should have been for what we needed at the time. And so they really diverted a lot of energy to make sure that they had the right focus. So that's amazing.
Yeah.
But these clinical trial sites is the other piece,
Yeah.
where this is recruitment. I mean, maybe you can just again give the sense of like what's a traditional recruitment time.
So I think vaccine trials on the whole can run very, very quickly. But what was so remarkable this time was that the clinical trial sites were trying to do this under social distancing. So if you thought that you'd be able to bring in 100 people in a day, but everybody had to be double masked and you could only have one person walk through the door at any one time, and then you had to go and sanitize the door, that makes things very, very different. And so running pandemic clinical trials is a very different experience for everybody. We were also very fortunate under Operation Warp Speed in that we had the support of the US Army, and they have incredible logistic capability. And so they helped us look at all of our sites, and where possible, they would come along and put a mobile unit into a parking lot and say, well, we can go and screen people in there and then they can go into the site. Things like that, things that we would normally have had access, well, nobody would have had access to that. They made available to us. And that meant that we could expand the footprint of every clinical trial site and hospital. Also, the diversity of the population that we were enrolling was incredibly important. We wanted them to represent the US population. And so they also brought in, we were able to create satellite sites in different communities, take the trial out to different communities around different cities to make the trial much more accessible to different populations, so that people weren't having to try and travel into a city to attend a hospital, but could go out and partake in the trial locally. And that was something that was quite different from the way many trials are set up, where people are prepared to take longer and everybody goes into a traditional trial site. So I will say that COVID-19 has completely changed and revolutionized the way I think we do a lot of clinical research. And that's partly because we had to look into all of these new opportunities that the industry had been waiting to sort of really do on mass scale. And then all the vaccine companies were like, okay, let's make this more accessible, more open. I think Jagesh, before he disappeared, asked me the question around how do you find, I think he started to ask a question about finding people to be in these trials. And he, one of the things was people were emailing us every day going, can I come and be in your trial? I want to be part of this. I want to volunteer. And it's a 2-year commitment going into this trial because we look at safety data sort of long term. And I can't say enough. I know there are some people in our trial from the Lexington community, because I've heard people sort of talk about their experience. Just heroes, just absolute heroes. You've got a 50-50 chance of getting the vaccine versus getting a saline injection in that trial. And then to sort of go and participate and volunteer and be part of history is just an amazingly altruistic thing to do for the benefit of research. And particularly as we actually didn't know mRNA was going to work. When we started, to be honest, I think there was a little bit of kind of BioNTech, Pfizer, Moderna, can you kind of get out the way? You're done now. AstraZeneca, J&J, those are the proven vaccine, those are the proven vaccine technologies. This mRNA thing, we're not sure it's going to work. Can we just, get out the way, and then we'll see where the others are. And we're all very aware of the expectation was that there would be about 5 or 6 vaccines in this pipeline. And the fact that the mRNA vaccines, both of them, came out with such incredibly high efficacy, people had dreamed that it would be like that, but there were many who thought it would never be that high. And it's just been an incredible part of this story that the first two vaccines to go into clinical testing came out with such incredibly high efficacy. And I think that gave everybody hope that, there was going to be a vaccine for this pandemic.
I do think that to just riff off of the the mRNA question, it is true that what we've seen is that there's a variety of other vaccines that were based on kind of what I would say more tried and true technologies, the inert vaccines or some of them. And as you pointed out, this was a huge win, not just for, understanding that we could, stave off the pandemic, but now this technology now has been proven in a way that's very exciting. I mean, there are a remarkable number of opportunities for vaccines down the line, and the speed at which we would be able to respond to them. So I think that is going to be, again, I think it will be an ongoing story. It's just the beginning right now that I think that we'll see developed. I did want to make sure that we got to some questions.
I was just having a quick scan.
Yeah, I do think that I don't know if we got to the what is lipid nanoparticles, LNPs.
And
that's an important question.
It is. And in order to get the mRNA into the body so it doesn't degrade too quickly, you wrap it up in a lipid nanoparticle, and quite simply, it's fat. Lipids fat, and it just it kind of gives it this little cushioned bubble, and that allows you to that gets it into the body. It sort of protects it a little bit like if you take a pill and there are sort of various excipients and you have slow release and all the rest of it. So it's just the equivalent. It's just the vaccine equivalent of how you would take any other kind of medication.
Yeah, and it's again, as you described, it's the software of the cell. So that lipid actually gets it into your cells. The cells make the protein, and then your immune system responds. So it's in many respects, it's very surgical. It's just that protein. And I think that that's another piece of it, which is choosing the right protein. And you
absolutely.
Guys were able to identify the spike protein as a critical element. And for those of you that you're familiar with the flu vaccine, this particular kind of protein called a fusogen is actually the thing that a lot of our immune system responds to. So this is a remarkably intelligent choice of the right protein to go after. I did want to get at, there was a question, and this is maybe a more broader question. Somebody asked specifically around how you got to 100 micrograms, but I think the dose selection, and particularly in the context of how quickly you had to make that choice, that's worth exploring a little bit, I think.
Yeah, I will never remember that Friday. Actually, it was a Friday afternoon when the data came in, and it was a combination of understanding the reactogenicity. So I mentioned reactogenicity before, but that is what many have experienced having had any vaccination. You'll get, you look for, is my arm sore? Do I have a fever? Do I feel achy? Are my glands swollen? All signs that the vaccine is doing something in your body and your immune system is kicking in, and that's the reactogenicity. And so we studied 50, 100, and 200, and we had a look at what that reaction looked like, and then we had a look at some early studies that looked at what the immunogenicity looked like. So what kind of antibody levels we were starting to, we could predict we could achieve, and what we would see, what kind of reaction we would get, that, and really at that point, how protective that would be against infection. And so it's a balance between what you think will be will give the right response and protect an individual versus the reactogenicity. And so 100 micrograms was chosen because it was that right balance. And also, I saw another question in the chat about the dose versus Pfizer. Pfizer's is 30 micrograms. It's, you know, it's a different LNP and it's a different structure. And ours is 100 micrograms. And that you can't compare them side by side. You can't say they're exactly the same because there are some differences between them. But that was just a dosing decision that we made.
But it's critical, right? It's that critical question. And where do you do this kind of dose kind of ranging, as it were? That's done, you know, kind of early? Is it done, you know, historically it's done preclinically, but in a pandemic, what do you do?
Yeah, in a pandemic, that was the second clinical study we ran in May. We dosed some people with 50, some with 100, some with 200, and then we looked at the data. This is all human data. This is human safety data. And we looked at, that was essential to collect that human safety data before we started large-scale clinical trials. And, you know, as I said earlier, the difference is these are all the same steps that you would have always done. We just did them really quickly. You know, we have the same data to make the same decisions we would have done if it had taken us 10 years to do this. And I sometimes feel I can't say that enough, because that's the most common thing I see is I don't trust it because it was done so quickly. And it was, you know, it was all the same data, just done much more in parallel.
Yeah, excellent. So somebody asked, and this is an interesting question as to, you know, if I participate in a clinical trial and get a shot, do I need to do things differently to test the efficacy of the vaccine, such as not wearing a mask?
Oh, that's a really, really excellent question. To be in our clinical trial, you needed to be at risk of infection. So that we wrote that protocol in May 2020. And so being at risk of infection in May 2020 meant that you were somebody whose job meant that you were likely to be exposed. So for example, a healthcare worker, or you worked in the retail industry, or you were a teacher. So we had a lot of people in education in our clinical trial. It also meant that you were, we also looked at people who had comorbidities, so other diseases that put them at higher risk. So about 25% of our trial, we wanted to have people that had, at that time, we knew that there were risk factors for getting more severe COVID. We still know about them. So, diabetes, COPD, heart disease, other, diseases that put you at higher risk. And so we wanted to have, and we also wanted to have a good spread by age. So people were either over 65. We wanted lots of people over 65 because we knew that they were most impacted. And then, last but by absolutely no means least, we wanted the population to be diverse and represent the population of the United States of America. And so when you look at all of those factors, so we wanted a spread of ages, genders, obviously, to be equal, then the comorbidities, as well as a diverse population. And so we needed to identify clinical sites around the country that could identify the right participants. And then we also needed to then provide information that would make people feel informed about coming into the clinical trial. And so when I tell you that up until the COVID-19, the COVID-19 vaccine trials, about 94% or maybe 95% of clinical trial participants were Caucasian in the US. Yeah. All white participants. And our trial enrolled a population representative of the US. So 37% from communities of color. So I think that's something that I'm really pleased that we were able to do in that period of time, because it was so important that the vaccine trials represented the people that the vaccine was for. Yeah,
and you're doing it on an accelerated schedule. That's even harder, right? To be able to make sure that the population comes in. I think so I'm going to go to a fun question, because some people ask a little bit, what do you do for fun when you aren't developing new medicines
or getting a new PhD or on planes?
Well, at the end of the day, I have to stop and thank my incredible family. They fed me, sometimes 3 meals a day. They completely took care of the house and the laundry and everything that needed to be done. And that's the same for pretty much all of us on the team, right? We were terrible husbands and wives and mothers and fathers and friends for this period of time. We could do nothing else. And our families held us together, and they were completely incredible. We were working 7 days a week, often 16 to 18 hours a day. And I don't know that there was a great deal of sort of time for fun. But I did, manage to get downstairs to see the family, and my husband every night would like put a small glass of wine in my hand, and I would probably fall asleep on the sofa about 5 minutes later. But I think I'm incredibly grateful to them, because I actually don't know what I'd have done without them. I don't think it would have been possible. So they are also amazing.
Well, that they knew what you were doing, so it's a remarkable thing to have people, support you in this, remarkable time. I mean, we're looking for support just making it through the pandemic, let alone making the medicine that's gonna, hopefully stave off some of the biggest issues. So I'm gonna still go to some of these questions. Some of these are gonna go and start getting to your basic science understanding. I know that you can handle it. Why?
Can I?
Sure.
Sorry, there's one. I keep seeing a few questions come up about variants, and can we just make sure that we get to the variant question, because I think that's a real interest.
So I haven't seen that one, but absolutely. So why don't we specifically go into the idea that this is a virus that's mutating over time, and that you're using a spike protein, and what is, what's the strategy that was generated early on that now you feel has been, how's it performing against the variations that we're seeing?
Yeah. And so thank you to the people who I've seen sort of asking questions around variants coming through, because I can completely understand why that's top of mind for everybody. So we knew it would mutate, because all viruses do. So if you think about the flu vaccine that you take annually, flu, there are new strains of flu going around the world each year, and a decision is made sort of in the winter in the Northern Hemisphere about what strain of flu vaccine will be available to vaccinate people end of every summer, and it's 6 months out for the Southern Hemisphere. So you have Northern Hemisphere and Southern Hemisphere flu vaccines available. And COVID obviously was going to go and do the same thing. SARS-CoV-2 was going to go and mutate as it went around the world. And so we saw Beta, and then obviously Delta, and then now Omicron as the main variants of concern. We've actually developed vaccines against all of them, and have put them, and they're working their way through clinical trials. And this is part of something that the world is grappling with right now. What came out of the G7 summit last summer with the G7 leaders is, how can the world be protected against the next pandemic? And so we have been looking at mRNA technology as, what can you do in 100 days? How far can you get? And so as soon as a variant of concern is declared, we look internally at, should we be developing a variant vaccine? And so we've developed that we've made combination vaccines with Beta and Delta together. We've made vaccines that look at the original vaccine with a variant. And that's one of the great things you can do with mRNA technology, is that is just a matter of days to put that together. And then you make enough to go into clinical testing. So I think people are asking how we're responding to variants, and we're doing all of that in parallel to watching it play out. So if you look at how Omicron is playing out, it's obviously highly transmittable. It's working its way around the world. There are some countries like South Africa which have peaked and come back down. Potentially the UK is in the same situation. And then, it came here after. So we'll watch with interest. And then it's, you know, the world health leaders around the world are looking at what kind of future vaccines do they think we'll need to boost a population responding to variants, if any. We don't know. I just want people to be reassured that people spend a lot of time thinking about this to protect us in the future. And that's where our technology is being really tested, to look at how we can do that and be prepared. And that's a huge amount of our work right now.
Yeah, and actually, I mean, to that point, there's another question in the chat which you can speak to, which is, how do you respond to a particular variant? Like, what is the kind of the difference between kind of the Omicron versus the Delta vaccine?
Yeah,
explicitly.
And that's the mutations on the spike protein, and it's sort of how the number of mutations and how different they are from the original strain that determines whether we're likely to see, you know, escape like we have done here with Omicron. It has a large number of changes from the original virus that we saw going around the world. And the impact of that has been, we've seen greater transmissibility, and you know, and also we're seeing increased breakthrough in vaccinated people. What we're also seeing is unvaccinated people have a much higher chance of being hospitalized and having severe disease. So a little bit like a flu vaccine, where some years in the flu vaccine, people will still, you know, feel unwell and get some kind of flu. It's nowhere near as bad as if they hadn't been vaccinated at all. And so I think that's what that's the picture we're starting to see with Omicron.
Yeah, and that again, it's the, you know, we don't want to make it too simplified, but this idea of just typing in a sequence and generating the vaccine is, I think, one of the keys to speeding up that first part of the drug development process, which is really, you know, understanding what your actual product is. I wanted to just, you know, in the last little bit, I wanted to really help people understand, you know, what is the process of the vaccination. So why does your body, under the expression of the spike protein, why does it even generate these antibodies and not against the rest of them? So this is a basic immunology question, but I think an important one.
Sorry, I missed the first part of the question. Why does it generate antibodies against the spike protein?
The spike protein, but, you know, clearly your body is not generating antibodies against the rest of, you know, what's unique about the vaccination? The fact that,
Well, what's unique about the mRNA vaccines is that you're only giving the body the code for the spike protein. You're not giving it any of the virus. So other vaccines will give you, provide you with parts of that virus that have been deactivated. But mRNA technology only looks at the spike protein, which so there is no other material for the body to react to.
And I think that, you know, again, what you were saying was that, you know, the spike protein is foreign, and this is exactly how the whole immune system works. So all vaccinations leverage this idea that your body can tell the difference between self and non-self, and the ability for the mRNA. So, you know, I think, you know, we're over time, so I want to be, you know, sensitive of, you know, kind of, you know, all the great work that you're going to be doing even over the course of the next few hours. So thank you so much for your time. This has been absolutely, you know, amazing to hear how quickly, you know, your team responded. Oh, by the way, they said they would only have 55 people. How many people do you have now on your team?
Oh, my team now is 300.
Yeah, so that maybe we'll have you back as to how do you do organization development going from 30 to 300 people. But I think that this really speaks to how quickly the company responded, and under your helm, you know, being able to make sure that, you know, we all have been able to, you know, benefit from the great work that you did. So I don't know, I can't thank you enough for what you've done already, and thank you for your time today.
Well, thank you. It's been a real pleasure to come and talk to you and the community. there are many people within the Moderna organization who are also in Lexington, but you have to understand it was a very incredible group of people who did a very amazing thing. And also a huge thank you to all of our families and friends who have supported us through a very crazy 2 years. We couldn't have done it without you. And I certainly have felt the love from the Lexington community over the last 2 years. And I'm very appreciative of that.
Well, thank you again. And best of luck. And again, thank you for your service. Truly.
Thank you. It's been great chatting this evening.
All right. Goodbye. And thanks,
Bye.
everybody.